Hi everyone, this is Elizabeth, and I am doing my presentation today on my microcosm project that I conducted by taking materials from the old Erie Canal State Park in central New York State. So, to give a little bit of an overview, we know a little bit about microcosms, and well, we know more than a little bit. But we know that they can provide valuable information about the larger ecosystem as a whole, and that experiments using microcosms have been considered pretty valid and very widely used throughout the scientific community. So, another reasons for doing this project is to figure out what's required to create a successful microcosm. And this was the site was located along New York's old Erie Canal, which had been converted to a state park in the 1960s. And I also, when I was doing this, thinking about kind of, as this is not, a naturally occurring body of water or anything like that, I was curious about whether this, their ecosystem in here would be healthy and robust and able to house and create a successful microcosm. So, going along with that, my first hypothesis was going to be that because this has been sort of a natural area for many, many decades now, that it should be capable of sustaining life. There's plenty of fish, plenty of birds, plenty of wildlife in this area. So, in theory, I was hoping I could get a pretty good microcosm out of it. And my second hypothesis was about the actual microcosm itself, and that after it was collected, we would kind of start to see some settling of different layers within the jar, and then that we would see some plant growth and maybe some invertebrates and some various life cycle processes going on, between the different living elements within the microcosm. So, to do this, I took my glass pickle jar, gathered sediment from the shoreline, water from the park, plants that were next to the water, and as well as log sheets and a pen to kind of record the information. So for my methods, obviously I traveled to the site location. From the shoreline, I collected sediment, water, and plants into the jar. Then I placed the jar on a shelf near the window in my office where it could get sunlight, and just monitored the progress for about 8 weeks to kind of see what would happen. So, starting off with the observations, the first week, as you can see from the photo on the left, it was pretty murky, pretty dark. Couldn't really tell kind of what was going on yet. That was right after collecting it. Over time, the water did clear up, but it kind of retained this yellowish tint to it, which we can see in the picture on the right. The plants that I did collect were green and had roots when they were originally collected. Unfortunately, however, they did not continue to grow once placed in the microcosm. They started to turn brown and decay and break down over this period of several weeks. The second week, we could start to see some invertebrates. So I identified fairy shrimp, freshwater snails, and there were some very, very other small, almost microscopic level of little things swimming around in here. But unfortunately, by the last week, there wasn't really much left at all. All the plants broke down. There's an explosion in invertebrates the first few weeks, and then slowly they started to become less and less as the weeks grew on, until eventually, by the end, there was not really any signs of life left within the microcosm. So that was a little unfortunate. This is an example of some of my log sheets. You can see kind of in the first one where a lot of stuff was sort of suspended in the water, still kind of just hanging out. As the weeks went on, it started to kind of, separate, settle out a little bit more. There was a lot of visible like movement of things we could see, which I suspect they were either in the soil or perhaps like eggs of some sort were in the soil and they started to kind of hatch during this time period. But then unfortunately you can see by week 7, it was much more settled, and the only things really left floating suspended, unfortunately, were deceased invertebrates. So the major findings here, my first hypothesis of whether or not this location would be able to support a healthy microcosm, I'm concluding that this is currently unclear at this time. The failure of anything to grow could have just been as much my collection methods and what was collected compared to the actual health of what was going on in this area. So some things that could have resulted in this would be a poor collection of materials. There was some algae and such floating on the surface of the water, which I didn't collect, but I'm sort of now wishing that I did. There could be poor quality water or other things in the water that may have been causing problems. There was not very much movement of the water. It was pretty stagnant at this location where I collected it, so it could be that something, toxic in the environment could have been staying in place, essentially, and kind of collecting in that area. There's also likely a good number of non-native plants and animals. I did see carp in the water, which I think are invasive, and there were definitely other invasive plants in the area. So these could be interfering with, what would naturally be living in this area. Another concern I had was maybe the sunlight from the window wasn't enough. It wasn't directly in front of the window, and we did have several weeks of massive amounts of rain and overcast weather, so I don't think a whole ton of light was always getting into this jar, especially at the beginning. So we can't conclude that the environment is unhealthy, because I could have just as easily not gathered enough plants or things that could live in this microcosm. And then my second hypothesis of that we would see some layers separating out and we would start to see some functions like plant growth, animal growth, signs of photosynthesis, respiration, nutrient cycling. I would conclude that this was partially true. We did see some things happening. We saw the emergence of the invertebrates like the fairy shrimp. We saw the death of some of them. We saw decay, breakdown of organic material. So some things did happen. Not fully what I was hoping for, but we did see, it didn't just sit in there with nothing happening for 8 weeks. Unfortunately, plant growth, photosynthesis did not occur, which I'm pretty sure probably led to the ultimate death of the invertebrates, because they didn't have anything unless they could eat decayed material, but there was also nothing in there to create oxygen either. So most likely this is probably what resulted in the overall failure of the microcosm to thrive. So some further research for this that I would potentially like to look into would be the actual quality of the water at this park. Is there anything toxic that's built up? Is there enough oxygen in the water already? So you can see in this picture a lot of these rocks along the shoreline, like those are not naturally occurring. So, but it also does look like there is a lot of naturally occurring, vegetation and stuff here too. So I'm curious to see, the overall health and quality of the water. Also curious about how the fish are doing In some areas, they seem to be, fine, but they're just, and this was just from what I could visually see. I didn't see any other fish other than the invasive carp. So it could be that there might be elements here that were not ideal for life. Along with the presence of invasive species versus native species, this would be kind of interesting to look at to see how many plants there are native versus what might have been brought in by construction work or other foot traffic. Also curious about the algae. There is a very, very thick layer of algae along the surface here. And in hindsight, I probably should have grabbed some of that for my microcosm, but I didn't at the time. So I'm wondering if that is maybe a large factor in what the invertebrates, can eat and what's possible to grow within the microcosm. So, and I also want to look more into the best collection methods and what to include and what not to include for next time, because I think that there were definitely some issues in the collection process as well as maybe where I placed it in regards to sunlight. So overall, this was still important. We still learned a lot because microcosms can be a really good study of larger scale things going on in the overall ecosystem. Quite a few reports have stated that microcosm experiments can be just as valuable as much larger scale experiments covering a broad area. We also learned small scale studies can be, used to easily controlled and manipulated to kind of examine larger issues. And this doesn't mean it has to be a microcosm in a jar necessarily, but it could be, in a lab, in a controlled environment out in the field, or something that's basically a small sample size of the overall larger system. We can do a lot of things like influencing the amount of light, the level of air, the level of water, the temperature, can all be controlled and manipulated fairly easily. And like in the example that I did, we can use it to study the overall health of a particular area, or if we want to analyze a larger scale issue, that can be done as well. And these are my references. So thank you very much for watching.